Okay, let's start describing the protein network. This protein network, of course, like anything else, it is working with several kind of interactions. But I can say that majority of this protein network is working at the post-translational modifications. You know what post-translational modifications is? It is adding or maybe removing a group permanently. It might be changing the structure of the protein allosterically, but all in all, it's the fast response. And we know that the kinases and phosphatases are very important in this protein network. Kinase is adding the phosphate group, phosphatase is removing the phosphate group, and these two are especially important to control the cell cycle. Okay, the very first thing to control the cell cycle is the cyclin CDK complex. Okay, cyclin is a molecule. It is a non-enzymatic molecule, and CDK is cyclin-dependent kinase. So as the name implies, it's a kinase, so it's an enzyme. It has ability to phosphorylate the downstream targets. Okay, when they were first found, they were found as the MPF, maturation promoting factor, or as I call it, mitosis promoting factor. So how it was found on the frog embryo. This is the frog as you know it. When you kiss it, it becomes a handsome prince. But this is the prince because it is laying eggs. And these eggs are giant. It is nearly 1 millimeter in diameter. So 1 millimeter for a cell is a real giant. And because it is so big, it can be investigated under the microscope. It was open to biochemical analysis, so it was highly used. So several things about the fertilization, about the embryogenesis, they were first started in the frogs. And what they have done is that they were getting a piece of nucleus here. They were getting this piece of nucleus with a needle, with an injector, and transferring it into another frog embryo. Maybe fertilized, maybe unfertilized. Okay, here. So what they have seen is that if the first cell is in the M phase, and when you inject piece of the cytoplasm into the next one, it is inducing mitosis proliferation immediately. Okay, the moment you inject this cytosol, actually it is not cytoplasm because you're not injecting any kind of organelle. The moment you inject a little bit of cytosol of this cell into this one is going under mitosis. So it is inducing mitosis. But if, for example, if this cell is at the interface, when you inject it here, nothing happens. It doesn't enter in the M phase. And the example is so glorious that even if this is an unfertilized egg, which is haploid and which cannot go under mitosis, and when you put a little bit of cytosol extract here, even this one try to goes under mitosis. Of course it cannot because the DNA is not replicated, but it still tries to. Okay, so there must be something in the cytosol extract which is giving the order of this cell divide. Sorry. So what is this order? Which molecule is giving the order? Because obviously it is not about cell shape, it is not about an organelle, because no cell shape or no organelle is transferred from this one to this one, only and only a simple mixture of chemicals. So who is giving this order? Who is giving the divide order? Later when they checked, it is actually something very simple, cyclin CDK complex and nothing else. So later on, they have identified this molecule in the clumps also. Tim Hunt made the experiment. These are the clumps. And this is a very simple model because it's easy. It's easy to keep alive, easy to take care of. And then it was also identified in the yeast. These are the unicellular eukaryotes. And one thing was very interesting. By the way, this is S. pombe, Saccharomyces pombe. This is Saccharomyces cerevisiae. These are the two different species of yeast. If you're cooking, if you're baking at home these days, you're quite familiar with the pombe cerevisiae. Okay, because this is what makes your dough puffy. Okay, this was quite interesting because the MPF they found here is the same with the MPF they found here. So from clumps to yeast, it was conserved evolution. And what was even more interesting was they took human MPF and injected into yeast, and even that was working. So MPF must be something so central, so important. It doesn't change between yeast and human. It is so central. So when they have checked the molecule, they realized these are the two molecules. This is the key. This is the molecule which has the visa for the passage from here to here. Okay, because in your passport you must have a visa. This cyclin CDK complex, which is also called MPF, is the visa which pushes the cell cycle from G1 to S, G2 to M, and between metaphase to anaphase. And everything else, they are just trying to stop the cell cycle from happening. Okay, when you check these two, this is a non-enzymatic molecule, you can see, and this is an enzyme. This activity of this enzyme can turn on and turn off. This is what actually the post-translational modification is. So when cyclin is alone, without any CDK, its activity is off. It's not active. When cyclin binds its best friend I'm sorry, this is a mistake. So when CDK is alone, it's inactive. Okay, the kinase activity, this enzymatic activity, is not shown. When CDK binds its best friends, which is the cyclin, then it is active. So without the cyclin, it is totally off. But all along the cell cycle, you can see that in every phase of the cell cycle, you have the same amount of CDK. The CDK level doesn't change. And people mapped the activity and protein level of cyclin and CDK, and they have realized that it is not the case for cyclin. This is actually why it is called cyclin, because in the interface, its concentration is very low at the beginning of the interface, which is early G1, and it is increased all through the interface. Of course, if the protein concentration, if the protein amount is increasing, it can increase with one single way, transcription and translation. So it is transcribed and translated from the genome. It increases. And here, when the cyclin concentration is low, it cannot activate CDK. Only and only cyclin concentration increases a certain level, it can activate CDK, and here you have this cyclin CDK's activity. After the mitosis, at the end of the mitosis, it decreases dramatically, okay? Very sharply. This sharp decrease is not because of entropy, is not because of anything else. Here, the cyclin is degraded. It sounds a bit brutal because it takes so much time to transcribe and translate it all through the cell cycle. You produce it for like days, and then in a single minute you get rid of all the cyclins by degrading it. And how do we degrade things? We know that the ubiquitins are added. It marks the cyclin for degradation. It joins the proteasome complex, which is a very large complex of degrading proteins, and then it is degraded in the cytosol. And who is doing that? APC, anaphase promoting factor, which we are going to see later. Okay, so all through the cell cycle, the cyclin level decrease and increase, decrease and increase, decrease, increase. This is the cyclin level. The CDK level is constant. The CDK level doesn't change. What changes of CDK is the activity. Okay, let's say this is the CDK activity. Just like that, the CDK activity is silent, silent, silent. Here it increase, decrease. Here it increase, decrease. Okay. The fact that you have CDK here in the interface doesn't mean that that CDK will be active. For activity, you need several other things. Okay, this is one thing. And another thing is that we have several kind of cyclin CDKs for different kind of for different phases of cell cycle. For example, here we don't have other cell cyclins. Here we have, I should use a different color. Here we have G1. Okay, let's call them here. Here we have G1 cyclin, and when G1 cyclin is combined with CDK, we call it G1 CDK. When you see G1 CDK, it's actually a combination of cyclin and CDK. Here you see the activity of G1 as CDK. So this is the passage of G1 to S. And right after the G1 as CDK, you're going to see the activity of SCDK. Of course, by SCDK, we mean S cyclin plus CDK. They are the same thing. And here we see G2M CDK. And here we see MCDK. So the graph I have shown here, it is the activity of the MCDK, not the other ones. The other ones have other graphs. For example, here you see a comparison of the S cyclin and M cyclin. This is inducing the activity of SCDK, and this is inducing the activity of MCDK. See the levels here. The level of S cyclin starts to increase early in G1 because it's going to be used here. And at the M phase, it decreases sharply because it is ubiquitinated by APC. The M cyclin is not produced in G1, in S. It is produced only in G2 phase. Here it increase, decrease again. Okay. The SCDK is responsible of pushing the cell cycle from G1 to S. The MCDK is responsible for pushing from G2 to M. Okay, so they have different localization and different functions. Okay, here we are calling it MCDK, SCDK, etc. This is the lecture, but in real life, when you check the articles about cancer, this is not what you're going to see. What you're going to see is other names like cyclin A, cyclin B, E, D, and they have specific CDK partners. And you see this combination and this specific point. So whenever you're reading articles, and you're going to read articles about the cell cycle soon, you're going to come across to these names. If you're not sure, you can come back and check this table. So let's